We have several programs actually running for developing treatments for Sanfilippo disease. What we know about these diseases is that they can cause significant neurodegeneration, so there is significant brain damage in these kids. And there have been 3 mouse models developed around 3 of the Sanfilippo types, type 3A, 3B and 3C. And really what we've been doing in our laboratory over the past 6 years or so is we've been trying to develop outcome measures that will enable us to understand how the disease is progressing in these mice, to see where there are similarities in the mice to the disease in the children, and to use these outcome measures to develop good treatments. So what we found is that in Sanfilippo A and B mice, and also in Sanfilippo C mice, that they recapitulate a lot of the things that we see in kids with these diseases. So they have neuroinflammation, they have a build-up of lysosomal storage of heparan sulfate, and also outside the cells as well, in the extracellular matrix and in the plasma. And they also have a very interesting set of behaviours, which behaviours are quite difficult to measure in mice. But what we do notice in these mice is that they're extremely hyperactive, and this is exactly what we see in the children as well. We've also noticed, we've published recently on circadian rhythm problems in the mice, and we've actually replicated this in patients as well. That will be coming out soon in publication. And so as a result of this, we've used these as many of our outcome measures in our preclinical trials. So we've developed 3 programs. The first is called a substrate reduction therapy, and we've used a drug called Genistin for this. And the principle of the substrate reduction therapy is that your body is normally producing these long-chain sugars, heparan sulfate. It's required outside the cell for a lot of signaling processes, and the body normally recycles this back into the lysosome, which is where proteins and long-chain sugars such as heparan sulfate are broken down. If you've got a block in that breakdown pathway, then heparan sulfate can build up. And of course the body is naturally producing heparan sulfate as well, in response to its needs for signaling processes outside the cells. So if you can actually reduce the net pool of heparan sulfate by blocking or reducing the amount of heparan sulfate produced, then you're achieving something called substrate reduction therapy, which is to block some of those production pathways. So Genistin is a soy extract, a plant product in fact. It's a fairly bioactive food supplement, and what we found was that Genistin works partly as a tyrosine kinase inhibitor. It also has estrogenic properties, and it's able to actually block some of the heparan sulfate production in these mice. So over long-term use, what we found by giving it daily to mice with MPS type 3B, so this is Sanfilippo type 3B, we found that we were able to reduce the amount of lysosomal storage of heparan sulfate by about 1/3 over a 9-month period. Neuroinflammation was reduced by approximately 1/5. And we also fully corrected the behaviour of these animals. So we found this really convincing preclinical evidence that we felt should go to clinical trial, and we're currently taking that through to clinical trial at the moment with an industrial partner who's providing drug, but actually it's going to be an investigator-led trial at the University of Manchester in the United Kingdom. So that was our first program really, and our aim with substrate reduction therapy was to try and slow down disease progression. We know that it's never going to be able to cure the disease. You're never going to be able to get rid of all of the heparan sulfate that's already built up. But what you might be able to do is to allow the cell to focus on different pathways of breakdown until it'll have to catch up a little bit by blocking production of heparan sulfate in this case. So really what we're after is a stopgap. Our second program is a gene therapy program. So what we've done is we've actually hijacked a technique which is available for other lysosomal diseases, for a disease called Hurler syndrome, MPS I. Currently, children with MPS I Hurler undergo a bone marrow transplant, commonly done in these children, and they see a really significant benefit from this particular treatment. Children with Hurler also have neurodegeneration, and somehow the bone marrow transplant is actually able to treat the disease in Hurler syndrome. Now, what we know about this disease is that if you provide replacement enzyme in the context of Hurler syndrome, then it can go around the entire body. It can be taken up by other cells, and it can be used by these cells to cross-correct the disease. However, enzyme is not actually able to cross the blood-brain barrier to any significant extent, which is why a treatment such as enzyme replacement therapy is not viable for children with a very severe neurodegenerative form of mucopolysaccharidosis type 1, such as Hurler disease, where there's no protein present. So, in a case like this, a bone marrow transplant is interesting because some of the cells from the bone marrow transplant can actually traffic across the blood-brain barrier. Now, we've shown that up to 22% of the cells that are derived from the bone marrow in the brain can be replaced in this way after a transplant. And we've shown that in the mouse model of Hurler syndrome. We also think the same is true in the mouse model of MPS IIIa, which is Sanfilippo IIIa. And that's where we've been working on a program of gene replacement. So what we've been doing is to take the bone marrow of a mouse with MPS IIIa, we gene modify it with a lentiviral vector to overexpress the missing gene, and we put it back into the mouse after chemotherapy conditioning. And at that point, we then look downstream. We've looked between 6 months and a year downstream. And we've done several different experiments using several different lentiviral vectors. And we always see the same thing. So what we found is that there's a threshold effect of around about 8.5% of enzyme that's required in the brain to actually see a really significant correction of neuropathological disease. So above 8.5%, and with the latest vector that we're using, we're getting approximately 11% in the brain, we see complete correction of the hyperactive behavior of these animals. We see a complete normalization of neuroinflammation. We see a complete normalization of the heparan sulfate storage and GAG storage in the brain. And we also see complete normalization of some of the secondary markers of disease that we've looked at, such as markers of synaptic function, so synaptic vesicle markers, and these kind of things. So GM gangliosides are another marker that's commonly used in these diseases. So all of these markers in neuropathology are corrected in this mouse model. So we are hoping to take this treatment through to clinical trial in probably 2 to 3 years' time. It's a technique that's been used quite extensively in the immunodeficiencies, Wiskott-Aldrich syndrome, and X-linked severe combined immunodeficiencies. And it's been very successful in those diseases. It's also been used recently in metachromatic leukodystrophy, which is another kind of lysosomal storage disorder in Italy. And we hope to replicate this kind of work in children with Sanfilippo IIIa. So our last program is on Sanfilippo IIIc. Now, this is a slightly different kind of disease from IIIa and B. And the reason for this is that in many of the mucopolysaccharide diseases, the enzyme is actually secreted. That means that it leaves the cell and it can be taken back up by other cells. This is the principle of cross-correction that we're relying on for many of these treatments. In Sanfilippo IIIc, the enzyme is not secreted. So it's retained within the cell. It's a membrane-bound protein within the lysosome. It still functions as an enzyme, so it's able to break down heparan sulfate in that way. The problem is that it's going to be very difficult to deliver it as a secreted enzyme. We wouldn't be able to secrete it from cells. And so we've taken a slightly different approach in Sanfilippo IIIc. In this approach, we're using an AAV gene therapy vector. We're aiming for a direct injection into the brain and really building on work that's been done recently in clinical trial, again, which will be presented at World later today, by a company called Lysogene in France, where they've looked at delivery, intracerebral delivery of AAV vector into the brain for Sanfilippo IIIa. So we intend to replicate this for Sanfilippo IIIc, and really what our aim here is to try and hit the cells that we really need to hit in the brain, because we're going to have to try and get as big a distribution within the brain as we possibly can. The brain is the main organ affected in this disease. So our aim in this program, and this has been funded by Jenna's Just Begun Foundation, is to really develop this preclinical program. We've developed outcome measures for the mouse model of MPS IIIc that it actually has very similar behaviours and very similar neuropathology to MPS IIIa and IIIb. Again, like the kids, has a very similar sort of level of hyperactivity behaviour. So we're currently about halfway through that program, and we hope in the next year and a half that we will have a preclinical proof of principle that we can then take forward to clinical trial for this disease as well.